Welcome back, everybody. This is the 7th lecture, so we're almost halfway through the semester for E340, 542. And today you are doing a lot of the presenting because you're going to be presenting your reviews and demonstrations of some nanoHUB apps. I'm looking forward to seeing that. And a little bit of a planning of your project. I know there's been some movement in various directions on project ideas, which is fine. And then depending on how much time we have afterwards, I'm going to be talking more about Hill rate laws. I'll talk a little bit about some of the kinds of problems you can address using network models. And then if we're lucky, we'll get to some simple exercises on feed-forward networks, which is a homework problem as well. And so this will be mainly talking about Herbert's chapters 4, 7, and 14 in Appendix E, as we were last week. And as always, I need to remind you that this is live streamed and recorded with the chit-chat gaps edited out. So today, again, we're going to be doing your presentations and then talking about Hill functions. And we'll do a couple of exercises related to thinking about networks, and then also, if we have time, an exercise applying this Hill function analysis to studying network behaviors. So I'd like to begin by turning it over to you. I'd asked everyone to look at an existing nanoHUB app. I'd handed out a set of suggested ones. And I'd asked each team to run and do a write-up of the nanoHUB app and then be prepared to demo it to the class. It's up to you whether you want to present your description of what you found first before you do the demo, or vice versa. And as I said, this is something you can do in teams. So I'd like to turn it over to you. Does anybody want to volunteer to go first on this?
I think Joel and I can.
Okay, great. Why don't I turn it over to you then? And thank you for being willing to lead off.
Joel, do you want to share and do the demo first?
Yeah.
Sounds great.
So our assigned simulation app was about modeling cancer population using radio and immune therapy for cancer treatment. What this simulation is trying to predict is if when we treat one site of cancer cells, if that can affect a site that we don't know, how that affects a site that we don't know, right? So it starts by presenting a little bit of the biological background of what is cancer, and then the role of an immune system in the body, radiation, what is the radiation on the cancer treatment. I'm just going to skip because I think other people also have to present their things. I don't want to spend a lot of time here. And what are the roles of immunotherapy? I can put this in the chat if some other people want to do it at the same time or explore on their time. Just give me a second. Yeah, so they presented the mathematical diagram following a paper. I'll mention the paper on the end because otherwise I'll have to keep scrolling back and forth here. And the way they did was very insightful for me and Sean on how we want to present our app too, because they did a bunch of plots, always letting the user to tweak some parameters and see how much that would change on the plot in real time. And that is a good way of doing it. One thing that I noted on my notes, because we were supposed also to criticize a little bit the app, is that I don't know if this is very informative. Calling just RT1 is hard for you to keep in your head what RT1 is actually doing, or RT2, right? Or C1 and D1, what are those parameters? It would be easier if it was just, you know, rate of growth for cancer cell 1 or something like that. It would be better with more description. Although the person that created this app did something like that on this print, right? To inform what other values there is the same here. And we can see a little bit of the changes in real time. Another thing is this carrying capacity for the tumor was explained as the tumor can grow only until a certain point, right? Taking in consideration the number of nutrients and other things that contribute for cancer growth is limited. The limitation comes from these values here. Although for me, it still feel a little bit just a value without any, you know, any unit to say, oh, is it volume? Is it, what is this actually measuring, right? Or I don't know. It's hard to just by looking at the graphs get some kind of interpretation. But the person does a very good job on showing and explaining what we were supposed to be looking at. And this is a great idea for our simulation too, that is guiding, right? Not leaving just the plots there, but also guiding the interpretation. And after that, you can go back and look. But since the you know, these apps kind of tend to be linear, I don't know if this is the best place to put the expectations. Maybe the expectations could be first, and then you can look at what the plots are doing, because then you have already in your head, right, what you should expect. Moving on, and another good point on this project is that they start adding one thing at a time to the model. So on the first model, we're just looking at how the cancer grows, how fast it grows based on these parameters, the rate of growth and the capacity, the carrying capacity. And then we start putting treatments on it and see how that affects to the cancer growth. And it goes first with radiation, and then it moves to immunotherapy without radiation or radiotherapy, and then it unifies both of them, radiation and just radiation on the and looking what happens on the secondary part of tumor, and in the very end they do both of them, the immunotherapy and the radiation. Another good thing that I found was making the model available for us to look at and understand where how was it coded and how we should interpret this. Now that we, basically everyone that is in this course know a little bit of antimony, it makes easy for us to just, you know, glance at it and be like, oh, okay, I understand how the system works. So I think that's basically, oh, here's the paper, by the way. I thought I said that I would mention the optimal radiation dose to induce robustness system anti-tumor immunity. It was a great paper to give us ideas, most definitely, and also think about some of the protocols we want to follow when doing these apps. You know, how already before we start any coding, think about what are the parameters we want the user to adjust, what type of conclusions the user could get, and things like that was very important for us to think at the very beginning of this project. So yeah, if anyone has any questions, feel free.
I think that was helpful. One question I had, so you felt that they didn't explain the concept of carrying capacity clearly enough in the
Yeah, I mean, it is understandable what is carrying capacity. I will scroll a little bit fast, just a fair warning. Yeah, it is, you can understand what they are saying, but I think the lack of some unit, I don't know if it's because it's of the volume of the cell or if it's just actual nutrients, because we learned that the growth is also limited by the space where cells are. So that could be a factor for the carrying. It's understandable that it's also very generalized, right? If we add everything too complex, it could be difficult to understand.
So I had one question, and maybe it's in the app or not, but their fundamental thing that they're evaluating is if you're treating a tumor that you've identified, what happens to tumors that are not identified?
Only if you have, yeah, if you're doing both treatments with radiation and immunotherapy, it seems to be the best of all. Radiation on itself cannot really affect the other site.
Right. So my question is, why is that an important question?
Why is that an important question?
Right.
Because when you have a diagnosis, this is my guess, right? When you have a diagnosis of cancer, it might be obvious that there is one site, but you don't know if this cancer migrated already or is it developing in another place that is not identified yet. So
Right.
right.
So do they make that clear? Because that's really the key to this, is the fact that radiation can only treat cancer tumors that you found. And by the time you found a tumor, the likelihood is that there are many metastases that you can't find, in which case killing the primary tumor doesn't save you because the metastases will grow and kill you. And so the hope here was that if you irradiate the primary tumor at the same time as an immunotherapy, the induced immune response to the primary tumor would be sufficient to kill the secondary tumors that you haven't been able to spot. And so that's really the key value in this work. And I'm just wondering if they make that clear enough in their introduction, because if you didn't mention it when you brought it, when you introduced it. And so I wondered if that was an idea that maybe needed more emphasis, because that's really the reason it's an interesting question.
Right. There is a little bit of a mention in that, but I think having like a conclusion here on the bottom, just to make sure that the point is clear for everybody, because when you're reading on the introduction, you get lost on the amount of information that is given at the same time, right? What is the concept of cancer and everything like that? It's mentioned there a little bit, but I think it get lost on the multiplicity of information. And another thing is that the models don't actually show things going back to zero, except when you put everything. I don't think everything maximum even. If you put like a lot of days, you see a recurrence of the cancer back to the same level as before. So I don't know how that was supposed to work, if this is what is expected, because you have like if you do the maximum radiation, for example, and then if I'm not wrong, lambda is, I forgot what lambda was here in this part. Yeah, the efficiency of the immunotherapy too. It's interesting because the cancer also comes back after X amount of time. So it's not saying, this model is not saying if you do both of the therapies, it will get cure from cancer, right? They don't claim that either. And they're showing that on the model.
So
So-
one thing maybe they could plot would be the amount of time it takes for the cancer to recur
Mhm.
versus the therapy. So you could ask the question, even if you don't cure it, can you keep people cancer-free for some amount of time?
Yeah,
And then try to understand how that time depends on the treatment approach.
yeah. Yeah. But it's a very good, very well done model. And I like the interaction that we can have with it that gave us ideas of how we're gonna implement on our models too.
Take that as a segue for me to start sharing. Hoping someone can just confirm that they're seeing my screen. Thanks, Joel. All right, so yeah, Joel and I are planning on doing this cornea stem cell differentiation models, trying to demonstrate homeostasis with the introduction of wounds. And so just to provide a little bit of biological context for that, I know Joel's already presented on this a little bit and has created these nice pictures here, but you know, stem cells proliferate and differentiate. We're going to try to capture that in our models. Where it's population dynamic models that we're worrying about. So we're going to try to, you know, as a proxy for homeostasis, we're going to try to stabilize these populations. And then once we start playing around with the differentiated cell deaths, we're going to have two different ways of those cells to die off through just like a decay function. And then we're going to allow the users to introduce these simulated wounds. And just something I want to call out, hoping that you guys can see my mouse pointer here, but in this diagram, these red egg cells are the stem cells. We have these green boxes are these TAC intermediate cells. And then these other three cells are the differentiated cells. And that'll really come into play in the next few slides here. But we have stem cells, intermediate cells, and then three types of differentiated cells, some of which can turn into, differentiate into one of the others. So for the project, the scope statement that we came up with, a little wordy here, but I'll just read it. It's simulate the wounded cornea stem cell cycles by establishing homeostasis in various topologies of restricted minimal regulatory models. A lot of big words in there, and so I'm just going to break them down a little bit. So as I said before, we're kind of doing this homeostasis. We're going to proxy that via stabilizing population dynamic models. Whenever we say these various topologies, we've identified roughly about 100 topologically different models that we're hoping will or should exhaustively capture all the permutations of a restricted minimal regulatory model. And we're kind of stealing that terminology from one of our research papers, kind of the main paper that we're hoping to extend on. And what they mean by that restricted minimal regulatory model is they're imposing an assumption on the models that there's a minimal number of regulators. So if you have 3 species, for instance, each species has only one regulator. And in fact, they showed that in order to stabilize under the conditions of these minimal regulators, each species has exactly one regulator. So to kind of just demonstrate that a little bit, because I know it kind of can just get lost in the words, these 20 different models, you don't have to worry about a particular one, but these exhaustively capture the minimal regulatory networks that would allow for stability in a population dynamics model. And as you can see, each of these have exactly one regulatory link coming out of each species, just in different, topologically different ways, depending on what they're activating or inactivating. And so I'll get into it in a bit when we start getting into the milestones, but just so it makes a little bit more sense, I'll get into it just a little bit here. We have a 5-species model that I showed previously with the stem cells, the intermediate step, and then the 3 differentiated cells. In our first step of the project, we're hoping to just kind of group all 3 differentiated cells into a single collection that we'll just name differentiated cells. And so we'll be able to create these 20 different topologically different models of stem cell to intermediate cell to differentiated cell. So that's really the first milestone. We're expecting to just spend a week on it since we know the exact models that we need to implement. So it'll be these stem to intermediate to differentiated cells just collectively. After we create those models, we're hoping to show that they can stabilize, which the paper was saying works mathematically. So no concerns, hiccups there we're hoping to hit. So we're thinking a week for that should be sufficient. And then where it starts to get a little interesting, I'm actually going to go back to the slide here with the different models. In these models, each of them on the intermediate species, it's able to proliferate. That's the symbol here, just means like splitting, proliferating. And then it does its differentiation. Once we get to our 5-species model, our intermediate species is going to be this basal, I believe it's basal, maybe it's wing, but it's one of the differentiated species here. It doesn't proliferate. It can only stay itself or differentiate or decay. And so we've identified out of these 25 models that would accurately capture the relationship that we're hoping to model here. So if milestone 1 is to construct the 20 models with 3 species, milestone 2 is going to be to turn that group of the collective differentiated cells. We're going to introduce the 3 specific ones, and how we plan on doing that is to take these 5 identified topologies and incorporate each of them into each of the 20 models. So at the end of this milestone, which we've dedicated at least 2 weeks to, we're hoping to have 100 completely unique, topologically speaking, models. And we're hoping that each of them show stability. And then the kind of last milestone, at least in terms of the project, would be to introduce a way to perturb these models. And the way that we're hoping to do that is this like simulated wounding effect. So for each of those 100 models, we will have some parameters that a user could tune to simulate an extended death of specific model or specific species. And maybe because the timeline works out that that's roughly where the mid presentation and report should be, hoping that we can, maybe speak to some of the models, some of the outcomes that we've noticed just from playing with it. But I think we've extended, or we've planned for this to last a little bit longer than just the midterm point. So we may not have, 100%, wrapped up. The final milestone being just to kind of put a bow on it, make it into a nice nanoHUB project. As Joel was saying, we're hoping to kind of utilize those sliders to allow the user to specify wounds, and in doing so, specify the severity of wounds and the specificity of which species they're hoping to wound. So that's the project. Open it up for questions, comments.
I think it's nice. One thing you might think about is, I mean, replicating some of those um, phase diagrams that were done in that source paper of Natalia's, where she shows for different networks what the stability boundaries are.
Yeah, we
Those-
were talking exactly about that. We didn't know if we want to do with like a vector field that I know Sauro teaches on his book a little bit, right? And I think Connor also already made some use of it, so I could learn with him how he did it. But yeah, it's true. That's easier for us to analyze, because you're correct. Although there's 20 here, not all of them lead to the same type of homeostasis. In some of them, we have like a runaway of stem cells, for example. In others, it's the TAC that runs away. They're still stable because there is a continuous number of cells. So that's how she says that is homeostatic. No cell die completely, but they are completely different. And also the stability part is very important because some of them sit on a very unstable equilibrium point. So any perturbation to that value put the whole model away from going back to a homeostatic situation. So yeah, it's true.
Great. I think the challenge will be to organize all of those possibilities in a way that the person using the application can understand. But I think you've got a great layout here for doing this. Are there any other comments or questions? I mean, one reason I think it's important for everybody to see all of these is because I think it's useful for people to see how other people in the class are thinking about these problems. And so it's perfectly okay for people to ask about how something that somebody else has presented, either what you understand or don't understand or clarification, but also if you want to think about how to apply it to what you're doing, that's fine too.
All right. Not hearing anything, so I'll hand it back to someone.
Okay, who would like to go next at this point? We have to get through everybody, so it's only a question of order.
I can
Lindsay?
go for Grant and I, yeah.
Okay, great.
Okay. Let me share.
Did we lose? Did we lose Grant?
I don't see him.
I thought he was on the call earlier.
Maybe it's possible that he lost connection or something like that. But we've talked about all this. So anyway, our nanoHUB app was this synchronization in neural dynamics one, which is just, the goal is just to give like a general overview of how neurons generate synchronized patterns versus unsynchronized patterns, and what does that mean in terms of the mathematics of these type of models. So it just starts off very basically building up the biology of neurons, how they generate action potentials, and the types of ion channels and things that they employ, how those work. And ultimately, all of that information is put into a model of cortical columns, which the equations for each one of these cortical columns have the same sort of form as an equation for an electrically excitable neuron, single cell. But the assumption is that each cortical column is made up of just a collection of similarly cells with similar properties. So yeah, the intuitions that you have gleaned from the background section before can sort of be directly translated into this specific set of differential equations. All of the parameters are really nicely defined everywhere. The equations are explained in a way that I think makes them very accessible. And then, like Joel was presenting, this one makes use of a bunch of interactive plots where you can kind of play with the parameters. And there are a couple prompts that are given in the text as well, just of things that you might want to look for or try. Like I think this one tells you to look for maybe a chaotic situation could potentially happen in this system. One thing that I would appreciate, I think, in this context is instead of just saying, look for chaotic firing, maybe give like the user some examples of parameter settings to try, because like in this example, I was unable to find a chaotic attractor. I mean, maybe that one. But yeah, it might just be helpful to give a little bit more guidance for some of these things. And then just like the representation of the same system in phase space, which is helpful. And then it's built up in a really nice way, complexifying it, where before we were talking about only one cortical column, now we're coupling two cortical columns together and seeing how that, because now the variable of interest, which is synchrony, comes into play. And so seeing how different settings of the parameters can affect the synchrony or rate of convergence in this case between the two cortical columns. And then finally, just increasing it to the 3 column case. I think it could have been a little bit more clear what rate of convergence was specifically, and also maybe other like measures of synchrony that you could consider, since the goal of the app is to talk about synchrony in general. I feel like maybe that didn't come across so well. But I thought everything was very well explained. And then a couple additional materials are offered, like background on dynamical systems theory and neuroscience and ion channels and all that stuff. Does anyone have any questions about the app before I just give a quick overview of what Grant and I are planning to do for our project timeline?
Well, I think I had a couple of questions. I thought, again, one of the things that's sort of a pity about this course is that people tend, it's always an option, but nobody ever seems to do it, which is to pick up one of the existing apps and improve it. Because a lot of these I think are very good. If somebody spent another semester working on one, it could go from being good to being excellent. But I thought you brought up a couple of good issues. Maybe it's something that Joel talked about a little bit and you talked about a little bit, which is how effective do you think this is as a learning experience? In other words, is it something that would appeal to people if you went through the text and did the exercise? Do you think that somebody coming to this would really learn something from it?
Yeah, I really liked how the background section started off just so basically. I feel like it would be a good resource for somebody who'd never seen these neuroscience principles before. I do think maybe it's missing a little bit of an intermediate step between this kind of background and then jumping straight into this rather complicated equation. There are a lot of like equations that have similar forms to this, which are much more common and much more easy to understand, with fewer parameters, like Hodgkin-Huxley, like Morris-Lacar versions of things that could have been helpful to add to this before throwing people into the deep end with this one.
So is the problem of synchronization explained clearly? In other words, why is synchronization an issue? Is that actually explained in the text? I think I appreciate the way that they're coming at this from a dynamical systems perspective, and introducing the idea of steady states and limit cycles and stuff like that. But I think part of what's missing is, like I said, different ways of measuring convergence or synchrony. And then, like you said, I think just another piece of connecting that back to biological significance. There's a little bit in here, I think in the beginning, or maybe at the end, about
relating this to epilepsy. And I think that would have been interesting to expand upon, but it's never really thoroughly explicitly linked.
I have a question. What are the biological responses when they are not synchronized, when the signals are not synchronized?
Basically, it's just like the phase of them get off. So you can have just the spikes that tend to not happen at the same time.
And that translates to some like, I don't know, like some disease or some other thing, like spasms or something, I don't know, in the brain.
It's kind of, it's two sides of the same coin, because you like need synchrony to produce learning and like processing and a bunch of other things. That's really important. But if you have too much synchrony, then it becomes like an attractor essentially that you can't get out of. And that's one of the things that's thought to cause epileptic seizures.
Right. It's a fascinating question, because excess synchrony is a disaster, is an illness, and yet insufficient synchrony is a problem. So you have to have this fine balance between the two. I think that's it's a great problem. I mean, one thing you mentioned more technically is they talked about chaos. I don't know if they defined chaos.
Yeah, no, the paper has a whole section on the types of chaotic attractors that you can get. But yeah, it wasn't so much in here.
And in principle, you could, I could see why they didn't do it, because it's a fair amount of work. But you could, in fact, create a plot that actually showed whether the time series was chaotic or not. So you could create a plot that either showed a return map that showed the transition from periodic to chaotic behavior, or actually calculated and plotted a Lyapunov exponent to show whether the time series was chaotic or not. Those are both more work. You have to do some mathematics to calculate those things. But that might have been something they, if somebody had more time, they could have added. So when you're exploring, looking for a chaotic state, you could do that.
Yeah, definitely.
The other thing that you mentioned, which I think is I don't know if they discussed or not, is that they show synchronization between two oscillators, and then they show synchronization between three oscillators. And I don't know if they discussed at all, but there are some fundamental, relatively profound mathematical differences between the synchronization of two oscillators and three. And so again, that may be, for one person project for one semester, that may be too much. But if we were thinking about this as a learning tool, it might be interesting to explore some of the history and questions about synchronization among multiple oscillators rather than pairs. Because back in the day, the mathematics of synchronization of oscillator pairs was done fairly early. But building an understanding of what happens when you have more than two oscillators actually took 30 years more. It was a very hard problem. And so it's, again, you have a limit to what you could do in one app. But when you mentioned that, it seemed to me that that's something that could at least be talked about a little bit, perhaps, in the text.
Yeah. Another thing that I thought could have, I mean, again, it's one person and one app, so it makes sense that it would have focused on this one model, but other like interesting neural synchrony models like the Kuromoto model and stuff, like I feel like could have made this a more complete story.
Well, was it useful to go through this for the things you're thinking about at all? I tried to assign the apps that I thought would be closest to people's interest, but I may have missed on those assignments.
Yeah. I think like especially the formatting of the equations and everything, and like the way they built up the equations is a good idea for how to make things clear to someone who's seeing all this stuff for the first time.
Yeah, I think this is one of the more elaborate projects that we had. I mentioned the student now is a PhD student at Purdue, and I think they put a lot of effort into it. So it's interesting to see your response. Okay, all right. So thank you. Any other questions from the audience? Again, the goal of doing these reviews is not so much for me to hear them, but for everyone in the room to hear them. So feel free to bring up issues that you think are important. Okay. Now, Lindsay, were you also going to talk about your plans, or
are we we're supposed to do that right after, or whatever's good for you?
I was originally thinking we'd go through all of
Okay.
the exercise, all of the examples first, and then come back to the plans. But Joel and Sean did it the other way, which is fine. So I guess no need for you to have to speak twice in a row, I guess.
Whatever is good with you is good with me.
So who's ready next? Who would like to present next? Danusha and Purva, you want to tell us what you, and Supriya, do you want to show us your review of the project?
Hi, just a minute, I'll just share my screen. Yeah, so is it visible? Anyone can say that. Okay. So what the model is the project that was given to us to review is basically a vaccine model for COVID-19. So what the project basically talks about is it's basically an educational tool to study what happened. It's basically a model to understand what happened in the United States, how vaccinations that took place in the United States happened. So this is actually a very interesting and a good project application that I came across. So it is based on the paper, which is this that we can come on at later on. And so it actually deals with three systems where scenarios where first considering that considering that people are not vaccinated when the COVID came, and then stepping into conditions where vaccination came, and then considering some other parameters too. So it's basically based on the model, which is SVIRD model. So that represents is S for susceptible, V for vaccinated, I for infected, and R for recovered and dead. So here's the model. So a susceptible person can either get infected or can get vaccinated. So after getting vaccinated, they can either get recovered or they can get infected again. And the infected person can either get recovered or dies. So this is what model says, and here are all the differential equations for that. So this application is actually really well planned. So here they are basically first examining the parameters where there was, they're considering the scenarios where there is no vaccine. So basically settling the bases. So where bases, and then after that, like here we can consider the case when there is no contact during the infections were taking place. But as we increase the infection rate, the contact rate, the infection is also increasing. And after that, the another case is the recovery rate. And there are set these basically first ones are where there is no vaccination injected to people. And then the second scenario that came in was when there is vaccination, people are vaccinated. So after that, and then the third one is considering the scenario where people are asymptomatic, so basically not showing any symptoms, but they are infected. So here, what we changed was, again, a susceptible person can get either vaccinated or go into the asymptomatic stages, and then after that, again, show the basis of infections, and the infected person can either get recovered or die. So here, not only the bases that were considered were based on vaccinations or the disease, but also the natural causes. Like some causes, the death rate can definitely happen for natural reasons also. So that this app is actually very useful in studying all the bases from beginning to then building up, like there is no vaccine, then vaccine is introduced, and changing a lot of the different parameters. And after that, it was also, so this app was basically made for educating people to understand how the system in US, the vaccination, when vaccinations were provided in US, resulted. So after that, here is some questions that were answered for this model. And the questions were, yeah, what is the fraction of population that needs to be vaccinated so that the infection rate either comes down, or people have like a very less chance of getting reinfected due to contact or something. And or if there is like a limited amount of vaccines, what should be the optimal amount where vaccines should be provided to the people. And also, again, is there a need for revaccination? So these were all the questions that were answered in this model. And I think that this model can help us in our project is actually by that we can start from the scratch, like setting up a very basic scenarios, and then slowly build up our a disease scenario, and then answering the questions for like what needs to be dealt and how can we deal with this disease. Yeah, that's all the project was about, I think. So I'm open for any suggestions or questions.
Did either of your collaborators want to add anything to that, Danusha or?
So yeah, this is what the model. Hello. So what, there's nothing much to add. She pretty much covered all the graphs that has been showed in the model. And then, yes.
That's okay. I just wondered. I don't know if what one question I had is, you mentioned that there were a bunch of things that were covered. Was there a single thing that you found most interesting, a single takeaway that you learned from doing this exercise?
So one thing that I liked is to see how the invention of vaccine directly, brought down the, increased the slope of recovered individuals and how it changed. So in the beginning of the paper, it's mentioned how the cases would have been if there was no vaccines. And then the second part of the paper, they mentioned how it changed after the vaccines were invented and how it was, how it became better when it was put into use. So that was something really interesting, which brought down the infected numbers, and then it increased the recovered individuals. After that, the one more thing that I found interesting was the third part of the paper, where they discuss about the part when people have to become revaccinated because the efficiency of the vaccines were not enough, so that they were becoming resusceptible again. So the susceptible population is increased, and then it decreases, and once again it increases. This is something that I found interesting about the paper, about the model. And yeah, it's a pretty good one. Like, it's good to visualize. Everything has been put clearly. They have added figures for all the models, which helps us understand the model better, even though we do not have prior idea about what it was about. So it's a very clearly demonstrated model. That's what I would say.
Okay, great. Other comments or questions from the audience?
I was wondering, is there any sort of like dynamical analysis that they do? Because I know at least the SRI model, maybe not this more complicated version, is like pretty well studied about its equilibrium points and how those depend on parameters and stuff like that. So I was wondering if you could do that in this model as well.
So in this model, I did not find a place where they used any equilibrium constants, but they have used like a lot of rate constants, like how the change is happening. But they did not mention anything about when it reaches equilibrium.
So you're saying that it might be, Lindsay, you're saying it would be nice if they had some plots where you had the x-axis was a parameter and the y-axis was the steady state values to get a sense of how effective the vaccination was. Or maybe even the x-axis would be a parameter like the vaccination rate, and the y-axis could be something like the maximum number of people infected. So
yeah, because I know that sometimes you can make plots like that with like the SRI model and say, oh, in order for us to not have an epidemic, we need this number of people vaccinated or taken out of the susceptible population, or stuff like that. So
basically do some parameter scans to explore the phase behavior of the model.
Yeah.
I guess one thing with those is that there's often, you can certainly make them have usable controllable parameters, because you could have two parameters that are the system scans, and for example have a third parameter that then is under user control. So when you change that third parameter, the phase diagram changes. So it's definitely possible to do that kind of thing. And I think you're right that in here that could be quite interesting. Because here, when you change a parameter, you have to remember what you saw before, right? It doesn't, you don't have a copy of what the curve looked like with the other values. So if you found something interesting, you'd have to make a note of it on a piece of paper, I guess, and then come back to it. That's a good point. Okay. Other comments or suggestions about this particular example? Okay, let's see. So who have we not heard from? We haven't heard from Peter and Zach. Do you have something?
I could go if I mean, I know mine's relevant to the past one that was just made.
Okay. Some of the ones to look at were sort of similar to each other, so it's okay to have some overlap.
Yeah. So, can y'all see my screen? Anyone?
Yeah, we can.
You can? Okay, cool. All right. So this paper, this nanoHUB project was also on modeling the COVID-19 pandemic, which I, don't blame them. It was a pretty popular topic for the past some years. But this one, and I think that the previous one also, so the previous one that we just looked over that prove a point it showed was, they called it an SVIRD model. In this one, there is a B on it, which I think on their graph, they also employed some sort of mechanism like this, but the B stands for birth. And so, yes, again, it is based off of the SIR model that we all know and love, but it is more complicated than that. Of course, the birth is basically specifying that the date that, there's a natural birth rate. So more people are born into the world over time. And when they start on the world, ideally, we'd assume that they're just susceptible, that they are not born infected or vaccinated, at least not with current technology. But then also an interesting aspect of this model is that there are decay processes as well. So not only do you have the deaths from the infected from COVID-19, but you also have deaths from infected from other natural causes, and natural deaths from susceptible people and natural deaths from recovered people. Also, despite what this graph may show, vaccinated people can also die in this model. That was just a slight misprint. And then obviously over time, like based off of all these parameters, susceptible people will either become vaccinated or they can become infected. And then eventually the infected people, assuming they don't die, recover. And then eventually, assuming they don't die, they will eventually become susceptible again. And so all of these different things have different decay rates. So nothing is really super stable in time. And so, yeah, these are just all of the equations. And if we just skim through all this, they show the first example that they give, where they use, they have D1 and D2. D1 is just the total number of deaths, whereas D2 is the deaths from COVID-19 specifically. And so we can actually adjust these different parameters to see just how many deaths can be caused from it. So if we really max it out, we could just, immunity duration, sorry, the other way around. Yeah, you can really just kill a lot of people that way. And I think that I believe that D1 is actually just referring to natural deaths, just because otherwise this wouldn't be possible. And so going on, they also do a comparison between total population and the over time as the total population decreases. If the infection is bad enough and deadly enough, and then they also compare each individual population over time, given different parameters that you can adjust. And so we can see here that R is the recovered, because if you have it bad enough, then this will happen. This does not. I don't think you can't really adjust any of the vaccination aspects here. But moving on, this graph they show the R naught. So for those that aren't aware, R naught is basically the average infection rate for any person. So if I infect 2 people and another person infects 4 people, then that's our only sample, then the average would be 3. So our R naught is 3. And so what you can see though is that actually R naught is not a stable thing. So initially the R naught can be very high, but over time, if enough people are infected, or enough people are vaccinated, then that's going to shrink really low down. And what people want is for an R naught to be below 1, because once you have it below 1, then the derivative is negative. So it's decreasing over time, the number of total people being infected. And we can see that here, that over time, because you can see here that the recovered people are very high in this example, that the rate, although initially high for R naught, the R naught will be very, very low almost immediately because of the recovery having a time decay aspect, because as that decays, the R naught's going to increase as people are moving back into susceptibility. And then they move on to prevented deaths. So they look at actually how if we adjust some of the parameters of vaccinating the population, how that can show us how many fewer people may die. So in this case, I moved the start time really early, but you could move it to very late, showing that the more time goes on, the amount of lives we can save doesn't really change because vaccinations aren't possible. But if we move it to 0 just to make it simple, you can adjust the efficacy of the vaccine. So if you had a very poor vaccine, there aren't a lot of lives saved as much. But if you make it 100%, then you can save a lot if you had somehow the perfect vaccine. And you can also adjust the duration of immunity as well as the number of vaccines per year. If we adjust it to a very low amount, obviously it won't really change that much. So moving on, they also adjust. They show some more plots explaining like the different, everything SV, IR, and then D1 and D2. And then we move on to the second model that they created, or that they, made them their own version of. And that is with a double vaccination, which I'm sure lots of people are familiar with. So this graph is actually improved, is actually fixed a little because now vaccinated people are capable of dying. And so essentially what's happening here is that now we have 2 stages of vaccination. And one thing that I actually just noticed is, oh no, never mind. I believe that all vaccinated people with 1 vaccination have to get the second vaccination, from my understanding. Yeah, so it doesn't account for people that were just forgot, but they are still capable of dying. And then everything else is basically the same. And so if we scroll down, we can actually look at, we have some more parameters to fool around with. One is effort, which when adjusted for, which is actually pretty difficult to see here, but maybe if we max out vaccinations, we can see it more easily, as well as the immune duration. Oops, that's not right. Let's increase efficacy. There we go. So if you adjust the effort to 1, then the amount of people that are, I think it's that the amount, sorry about this, it's the effort in which people receive both vaccines, I believe. I think that's right, because when you adjust the effort to be very high, people only receive 1 vaccine, whereas when you adjust it to be very low, that's confusing. I'm sorry, I may have gotten this one mixed up. I don't really recall how that one works, but the fractional efficacy is just the percentage of which the first vaccine is good compared to the second vaccine. So if it's, let's say 0.76, that would mean that the people in the V1 category are 76% as resilient to infection compared to the people in V2. And it's kind of difficult to see here because there's so many lines here, but you can see this little orange line right here. And then there's the green line, that's the V2 population. This is the V1 population. And so if we were to decrease the fractional efficiency, then we would see that the percentage, the number of people in V1 actually go down because they're more likely to become infected. And if we went the other way, if we made it 100%, then there's really no reason getting a second vaccine to begin with. So moving on, then we also have the, we're looking at prevented deaths from the double vaccination model. And again, you can adjust this as well, where if we adjust the fractional efficacy from one vaccine, then it's less effective because over time, because it takes so much time for you to be able to get out enough vaccines. And then this is the other amount. And that's probably what effort's going on about. And again, I'm sorry, it's completely slipping my mind right now how effort works in this model. But that's basically it. It's kind of just an extension of what was just shown, but just now you have a double vaccination model. And there's also a more complex decay process. Any questions or concerns or interests?
Well, it sounds like there were some typos that need to be fixed. Sounds like they didn't define effort very well.
Yeah, I remembered it when I first read it, but let me see. So effort is the value that represents the different approaches to vaccine distribution momentum. Okay, so yeah, it is. I think it's just the how much they try to release the vaccination, how many people they're vaccinating. Is that right?
No.
Okay, yeah, it completely slipped my mind during the presentation, but you know, can't remember everything.
I have a question, but it's more like to the whole class. This is the first one that we actually see the code. Do you guys see any benefit of having the code into the app?
Well, I personally saw a benefit from seeing the code because of the first graph right here. Because I thought, oh, they made a mistake because they made vaccinated people immortal. And that is not the case. When you actually look at it, you see that they do in fact have the death rate of V. They just forgot to put it in here. So I think that it is very useful to have the code just because it proves that what they did is actually what they did. And I could imagine examples where the opposite is true, where they put this graph out, but then you look at the actual equations going on and you're like, oh, that's actually not how it works, especially when you have these charts to fool around with. And it's just nice to have the confidence to be like, okay, well, I know that this is in fact what it's doing, what it's supposed to be doing, and it's more of a misunderstanding on my part.
Right, on that cell number 2, is there any other way for you to change any of those values there? Have you tried? Like, can you write something there? Just write something there. Type something, see it types. Okay, yeah, okay, that's good. Okay.
So I also think there's something to be said for in terms of like propagating more helpful models from an educational setting, right? Like if you look at a nanoHUB application and you think to yourself, oh, this is really good. I like how they did these plots and how they formulated this, then it seems helpful to be able to see the code present as is. And I can also say for sure, to Zach's point, that the opposite could happen. Eden and I found a bug in our model, and it was really hard to kind of figure out what was going wrong with the code not being very like as easily accessible. It took some more digging.
So maybe it's worth breaking in there. Usually the default is that the code is hidden, but if you go up to the bar, the task bar just above at the top of your here, go down one line to where the icons are. If you scroll across, you say where it says app mode, click there. So hit leave. So the default would be, so you normally the default is that you hide the code and if you're in app mode, you have the code hidden.
Oh,
But you could always see the code. If you go up to the top where it says edit app, that will display the code. So in fact, people have the choice, but it's a question of whether you want the default to be when they first go in, whether they see the code or not. But probably everybody should have it documented in the code itself, in the app itself, saying if you want to see the code, click the button that's labeled edit app, and if you want to see the app in its presentation form, hit the button that's called app mode.
If you add the app, does it change the nanoHUB application version of the app?
No, this won't overwrite the nanoHUB.
Oh, okay.
You'd have to create if you edit this code, you'd have to download it and then upload it as a new version.
I guess the addition allows people also to modify, in their own ways, but that's more advanced, as you said.
But yes, if you hit edit app, then you believe you're able to download it. You should be able to download it afterwards. Yeah, you see now you've got the standard Python Jupyter Notebook window. So if you go to File and you do Download, you can pull your personal version of it. So these are things that Giuliano will cover when he talks about uploading apps, but they're things that I guess you had to discover yourself in this context. But normally the expectation is that the code is hidden, but it's not a requirement. But there's something to consider, which is from the point of view of the user, is it more convenient for the default to show the code or not? I guess depending on the user background and interests, one case may be more, might be intimidating, or the lack of the code as the default might be annoying. So they're both legitimate strategies.
were there any other questions or comments?
I mean, one thing you mentioned was that maybe some of the plots were too busy, that it would be better to maybe split some of those plots into two so you didn't have as many lines on each plot.
Yes. Yeah, I think I prematurely stopped sharing, but yeah, absolutely. On the later ones, I think it would have had to be this one, which I don't think I can run now because I think it just or if I go to app mode, I think I can. But the yeah, that one in particular, it was really difficult because of the scale to be able to view. Yeah, this one, it was really difficult for you to actually see the death rates in comparison to everything else. And also it kind of made it difficult to, I don't know, it seemed like it was just kind of like proof of concept more than anything else, where it didn't really accomplish much. Because I don't know, I guess in some ways it's useful to be able to see how the general population fluxes as your deaths change. But when you have so many lines, maybe if it was focused on just the infected or just the vaccinated lines, then it would make more sense. But just because there's so many things, especially because the recovered and the susceptible make up such a high percentage of the population, at least in this example, yeah, it does make it kind of busy.
So one thing to do would be to let the user choose what time region and x-axis and y-axis range they want to display.
Yeah, that might be it's the paradox of choice, where it becomes really difficult to, if you add too many things, it might be overwhelming to people. It would be interesting, I think, especially along the x-axis. It would be interesting. The y-axis, I don't know how much you can really do with, just because it's going to try, like, you kind of want to have as much information as possible based off of the y-axis.
So maybe splitting that graph into two and having some of the values on one curve and some on another would be better.
Yeah.
Clear.
Yeah, that would be nice, just so you get an idea of the proportionality of everything. I kind of wish that maybe there is something like this where you can plot different lines, but each line is proportional to they're somehow normalized in a way. That would be
You
interesting.
could certainly do that. I mean, you could certainly scale each line by the value, by some reference value.
Yeah.
That's I mean, the other thing, thinking of what Lindsay was talking about in the app she was reviewing, is it might be useful here to have some suggested parameter values, because you have 4 parameters to change. And it might be useful to have some ones to look at that might be interesting, because having to look at all 4, slide all 4 is a lot of choices.
Yeah, and it'd also be interesting to see the what Lindsay also brought up, a dynamical systems approach to it. Because with something like this, at least I can't imagine it reaching equilibrium points, at least maybe very far out into the future, but like generally no. But it would be interesting to see maybe limit cycles occurring based off of the right adjustments to different parameters.
Yeah, I think that's an interesting one. I had made a note about that too. I mean, you showed some situations where the system comes to us equilibrium pretty quickly. In other cases where you have decaying oscillations. And I know that there also are situations where you get continuous oscillations, where the oscillations don't die off. And so maybe some bifurcation plots where you have the parameter on the x-axis and the steady state or oscillating values on the y-axis, instead of just time on the x-axis, would be helpful here.
Yeah, definitely.
And just knowing what situations lead to the extinction of the virus, you get rid of it completely, what lead to endemic, where the virus is present at a steady level, what lead to persistent epidemics where you have the oscillations. Those could be interesting extensions of a model like this. Was there a single takeaway that you got out of this application? You said it seemed more like a proof of concept than a finished application.
I mean, I just think that it was just putting out I think what I meant by that was that you're just making they're just taking the BSVIRD double vaccination model, and just kind of showing how adjustments to that works in terms of like what happens with the dynamics. I just thought that it maybe wasn't question focused as much as it was just showing you what occurs when you do have a model like this. So I think that's what I was mainly getting at.
Great. Other people's comments or questions? Okay. All right. I guess we have Connor and Eden and Peter.
I'm ready.
Peter, go ahead, please.
So this one is another infectious disease, this time tuberculosis. And the app focuses mainly on 3 different things, which are the overall incidence and epidemiological aspect of it. There's the effect of a vaccine, and then antibiotic resistance. But sometimes TB can resist antibiotics. So they have that same abstract here and a longer introduction as well, saying that it's one of the most deadly diseases ever, or most infectious one. They then provide the complete model. It's a rather complicated diagram, but they just say it'll be explained later on, step by step. The first thing they do is map the population in 4 different groups. So similar to Zachary's model, you have uninfected, infected, non-infection, and recovered, except this time they point out that they have to remove the birth rate because in all those charts in this app, it would just be impossible to really see what's going on otherwise. So this one, no birth rate. There's also the R0 statistic, which they put below almost every chart, which is it's the same concept of how many people are infected. So this background section, I wanted to point out one particular observation, which was they say their overall TB prevalence is 15%. This number is not outrageous when compared to the literature. About 70 years ago, it was 60 to 75%. That makes me mistrust this application a bit. It does not really tell me when this data was collected that it was able to be so far off. But calling it not outrageous to me, it's just kind of a way to say our data is off, but not that off. So one of the first scans they have is number of interactions per day. As you would expect, you have a lot more cases overall. And it's just useful to have some quantitative data in that regard. You then get to scan through the same parameter they just showed. So here the values will scan up for you, and it's this very effective divided bar chart. Here you use a slider. So you say you put up the number of interactions. Let me go to more midway values so it's easier to see. Okay. So this is easier. So in this case, you have a lot more infected people as you get more interactions. And the R0 changes as you use the sliders as well. Generally, R0 will not keep up with interactions per day. They've also pointed out that the steady states will become delayed or brought earlier based on various factors that they adjust to, or the parameters they adjust to of the application. The next one is resistance to reinfection, which basically just means if you already have TB, then what's the chance that you're going to get it again? So if someone has a really high resistance to infection, then the number of recovered individuals will be about the same as it was with 0%, but you have a lot more people that are just susceptible. So this is the main insight they take away. There's this kind of like dividing point here around 50%, where recovered people stops growing and starts to decrease. You get to scan through the same. And treatment efficacy is shown. In this case, you're talking about an antibiotic treatment, and the values again range from 0% efficacy to maximum. You get to change to show the treated people separately. Does not change amounts themselves, but some of them are split up. So infectious now becomes partly treated. So it's the same idea here, where if you have a lot more people who, or if the efficacy goes up, then we'd expect the number of treated people to maybe stay the same, but susceptible goes down. So we're showing the diagram again. It's a similar concept here of you get to split up the population. This model assumes that when you first start out at T0, you have everyone is susceptible, but people very rapidly start to become vaccinated. They don't really provide any data behind this. And I kind of question it because there are a lot of people who resisted the COVID vaccine. So to me, this doesn't quite apply as well. Then we get the same diagram that we've shown at the sort of back. So one of the main parameters for the vaccine are the vaccine rate, which is this one, and then below we'll go on to the efficacy of that. So again, it's kind of unsurprising that you have a lot more of the uninfected people as you increase the vaccine rate. And there's a certain point where you start to have the epidemic be wiped out. If you start, well, for example, with this kind of vaccine rate, you have the vaccine occurring after about 20 years. To me, with someone who did not have a lot of background, it was unclear as to why the epidemic starts to occur at 15 years. Why do we not have really any visible cases out here? So it's kind of unexplained. But if we do increase the vaccine rate, it not only decreases the overall number of people who have the disease or are recovered from it, but also delays it, so it's way out towards the 50-year mark where there's any kind of increase at all. This one is the same as before. I'm going to take a moment to look at my notes from there. Okay, so I did feel that this app was missing some insights in certain places. For example, they did not explain how there's a significant jump in the number of people treated once vaccine efficacy reaches about 20%. So what we see is, sorry, the overall number of susceptible people increases a lot as we increase the efficacy. Again, the same concept as before, but even though there's insights below every chart, I do want to say that overall throughout the application, they sometimes don't introduce charts too well, or don't really give a lot of insights for them. It's very easy to understand what parameters you're adjusting, but I often found myself taking away information from the charts, but not finding it in these summaries below, meaning that it's very easy to miss some information. The last section is antibiotic resistance. There's a much more complicated diagram this time. I also wanted to point out that they do show these with the tables alongside them. Presumably we copied from a paper or two, but they do not have any kind of text that tells you what are the most important things you should be looking at. It's kind of up to you to scroll through the app to get that information. So that said, so what I'm trying to say is that it would be helpful to have some kind of summary. So this case in particular is where I mentioned there's not a lot of introduction to a few charts. So let's say, let's look at antibiotic resistance, and they just throw a chart at you. This one is comparing the incidence of wild type and resistance. So resistant type TB, they both start around maybe the same, or sorry, resistance starts much less than wild type, but it very quickly overtakes the wild type. So in this case, we are shown that resistant type behaves a bit more differently than before, but it's kind of the same idea as the original model. So if we compare it, we do have fewer people are susceptible, and you have a slightly lower number of infectious people. So this section is kind of awkward to me. You do have to scroll up and down a lot to get the results. What I would have benefited from most here is a side-by-side comparison, or maybe even overlapping charts. So some of the last parameters include the efficacy of the treatment. In this case, it's the vaccine on the antibiotic resistant model. Here they point out that whether you have the vaccine or not, this here is not, and here is a vaccine, really makes no difference. These charts are about the same. So they have the vaccine for the rest of the charts. So this is what I mentioned earlier, where you have a wild type and resistant type, but if you increase the treatment efficacy, you get a more dramatic effect of resistant type overtaking and dominating over wild type much more quickly. I believe this is the last section where you're looking at patient treatment. There's 3 groups here. SAT, which means self-administered, DOPT, which means direct observed, or a healthcare professional gives it to the person, and perfect cooperation, which is maybe more of a theoretical way. They say the person is perfect in taking the antibiotic. Again, it might be better to have side-by-side, maybe not, or at least some explanation, because you have to squint to see that self-administered is just slightly worse than people taking the antibiotics under a healthcare professional. Then finally, if it's perfect, then you just have it ever so slightly better again. So this is where it ends. There's a references section here, but no further reading or discussion. I felt that the ending to this paper is very abrupt, and again would have benefited from possible some summary or some takeaways that would explain why it was beneficial to look at this application. My verdict is that I would recommend this app to someone who is unfamiliar with the topic, like an undergraduate student or maybe myself, but definitely not to someone who already knows a lot about epidemiology. Maybe the only thing you can get out of this is quantitative data, which you'd have to look at through the charts. Are there questions or comments?
Great, another very thorough analysis. I mean, there seem to be some themes here. One of which is that you people need to be clearer about explaining what you're going to see and what you've seen in these. Sounds like having, again, some target values to try would be good. This one does have the parameter sweeps that Lindsay was wanting. Right here, you do have the plots where you show how the system changes with a particular parameter change. So that seems to be a strength of this app compared to some of the others. I think your point about having sort of discussion and further reading and conclusions was interesting.
Overall, the app was very easy to understand and approachable. But when I looked at the one Prof reviewed, I saw they had a subheading that was something like, what's the level of efficacy we need to actually stop the epidemic? So coming out with some questions like that, where you're trying to define a certain threshold that you should be looking at, would make the app more useful.
Great. I mean, I think there are 2 things that I was going to say earlier, one of which, again, is a theme, which is people often talk about R0 as if it were a property of the virus, the infectivity. But of course, the infectivity is a function of the virus and the social environment. If we all wear masks, the R0 of influenza goes down by a factor of 10. And so in the textbooks, you'll see R naughts for published for measles, for mumps, and for other diseases. And there may be some theoretical maximum infectivity of a virus, but the actual infectivity in a population will vary with time. And I thought that was something that maybe could be explored a little more in some of these apps. There was a plot in the earlier one that I was going to mention with the Zachary that presented, which was it talked about the deaths with treatment and without treatment. And here you could also have deaths from TB versus deaths from other sources. And this idea of plotting in the earlier application, they plotted the curve, which was the deaths from without the disease or without treatment, and the deaths with treatment. It occurred to me that it might be more interesting to plot the difference between those two curves, what are called the excess deaths, which came up a lot in the COVID epidemic. But I think you pointed out a lot of things that could be improved here. Was it clear in the introductory material? I mean, why is the vaccine efficacy something you want to study in the case of tuberculosis? Did it talk about the history and the reason that that's an important topic?
No, and they did not define what is meant by efficacy. If there's any sort of qualities that go along with it.
Because tuberculosis is interesting because there's been a vaccine for tuberculosis for a very long time. And in France, for example, most people get the tuberculosis vaccine. In the United States, you can't get the tuberculosis vaccine. And that's a little bit unusual. Normally, when vaccines are available, they're used. People may or may not have the vaccine, but it's unusual for a vaccine to be offered in one place and not another. In the case of tuberculosis, it's because the vaccine helps, but it's not that great. In other words, it may reduce your probability of getting the disease by 25% or 30%, not by 95%. And so there was some argument about whether actually the giving the vaccine could make things worse rather than better. This kind of thing has come up recently in the case of the polio vaccine, because it winds up that the live polio vaccine can actually cause polio. And in a population where the risk of getting polio is very high, using the live vaccine was worthwhile. In fact, it could lead to secondary, what we call secondary vaccination. Basically, people who are vaccinated spread the vaccine. But in populations where the risk of getting polio was very low, the risk of getting infected from the vaccine was much higher than from getting the disease itself. And so there is quite a bit of interest in what happens when you have vaccines that aren't great or have risk associated with them. And so maybe that's something that could be up. See there, when you turn the vaccine efficacy up to 100%, then effectively nobody gets the disease anymore.
Yeah, I wanted to
disease
point
goes
that out.
away. And so maybe that could have been explored more effectively. You could have had a question like, if you could increase the efficacy of the vaccine from 30% to 70%, what would happen? Or how effective would the vaccine have to be in order to be able to get rid of the illness?
Exactly.
So that's a possibility. I think the text could have been clearly more explicit here. And then also with the discussion of treatment and resistance, the development of drug resistance to tuberculosis is a big problem. One of the reasons that you have that resistance is because you have to keep taking the antibiotics for a long time, years. And the antibiotics are pretty harsh and make you feel pretty sick. And so one of the big problems with tuberculosis is You start taking the antibiotic, you feel better. The antibiotic makes you feel sick on its own right. And so the moment you stop feeling you're sick from the TB, you stop taking it. And that's a recipe for inducing antibiotic resistance. And Denise Kirschner has done a lot of research on this, trying to come up with better drug therapy regimes. But that's something that also could be explored and maybe could be discussed a bit more here. I wonder one other question. Do you think that this would be more effective if it were not one huge monolithic application, but it were a series of smaller modules?
I kind of want to say the opposite. I think it's good the way it is. It's certainly effective to have things very consistent where you're always comparing generally 4 to 5 groups of people. But what I felt was missing was something that really combines all of those. Like what happens if you have vaccine and antibiotics? Give me a model that allows me to adjust all the parameters at once.
Is there a way to cluster modules on nanoHUB? Like, I guess like I'm wondering, because on one hand, that sounds like in the case of seeing a bunch of code that could be less overwhelming for certain applications that have a lot. But if it's the type of thing where it's like, go out and then search for this application to find the next thing, I could see that getting very unwieldy very quickly.
Well, I guess you could have a link to the next chapter.
That's true.
You could have basically an index button that took you to the next app.
Or if there was a way in the notebook to collapse sections so you can sort of like open and close them and still technically be in the same link.
I think that may be possible. I don't know how to do it, but I think you may be able to cluster cells in Jupyter notebooks. I know you could do that in Mathematica, where you can have a hierarchy, you can have basically an outline format. I don't know if that's possible in Jupyter.
I know it's at least possible in the whatever Google Colab, their version of the Jupyter notebooks, because Neuromatch Academy structures their lessons like that. They'll hide chunks of code. I don't know if that translates over to Jupyter notebooks. So I feel like it should. There's a pretty one-to-one comparison between those applications.
Well, not every feature. nanoHUB doesn't implement all Jupyter notebook features. I don't know if that's the case here. I mean, nanoHUB also implements some additional features that are not a part of base Jupyter notebook. So again, I don't know the default would be that if it's a base Jupyter notebook feature, it should be there, but there may be some exceptions. Other comments or questions? That was a lot of material to dig through. You did an impressive job going through a long application. Did it help you at all thinking about what you might want to do or about how to present it? I know you've been thinking.
Certainly did.
Okay. I guess Connor and Eden, did you want to present? I think.
Yeah, we can.
Do you want me to share?
I have it pulled up. Do you also have it pulled up?
You can go for
Okay.
it.
Alrighty. So can everyone see the Jupyter notebook? Okay. So this is a cluster of models regarding cell-to-cell spread of influenza. Some of them are the first one's a pretty simple base model. And then from there, they go on to talk about a model including interferon and delay factors, and then a model involving antiviral treatments, and then the last one with an effector cell response. So we will, I guess, I'll do the first part and then we'll just volley back and forth. So to start off, quick comment on the summary page, even though I've already gone past it. It was pretty brief. This notebook as a whole is incredibly thorough and offers a lot of background. There were certain aspects in the actual application description that felt a little lacking if you didn't already have a fair amount of background. But one thing that was really nice is that they directly linked PDFs of the two papers that they were drawing from for the most part, so that you could more easily access the reading material that they were pulling from. They also go ahead and show some of the data for a viral load in patients over the course of 8 days. So you can actually do a drop-down menu and look at the individual patients one by one, and then you can also go and see the average data, which is what they use to fit the parameters of the models. So the first model is just 3 nodes. We have P, which is our population of cells, I, which is a population of infected cells, and V, which is the viral load. The normal cells get infected at a rate that involves a general probability, the current number of uninfected cells, and the amount of the viral load. The infected cells release additional viron, is that the right word? I think at a given rate here, and then the virus itself has a clearance rate, and the infected cells have a death rate. So we have a system of 3 coupled ODEs. And they also give, this is just a screenshot from one of the figures in the paper where they show how they get the parameter values for this default model. I have slight issues with how they set up the plots in this model. It's not a huge deal. I just found some of the axes to not be super intuitive. And I also found that it's not as much of an issue here, but I usually prefer to also have the symbol that was representing this in the equation next to the slider bar. Otherwise, you're having, if you're reading it and trying to explore, there's a lot of looking down at paper, scrolling back up, reading the paragraph underneath to remember which one connects where. So that just removes an extra step. But it works just fine. You can change the viral production of an infected cell. And when we do, we see that we lose our normal cells faster, and we also get a steeper increase, followed by a decline in the amount of the virus in the system, which makes sense because it is directly dependent on the number of infected cells present, and both of these have a decay rate. Something that would have been nice is actually seeing the number of infected cells in this plot. I feel like that could have been added in a way that made this relationship here clearer. We can also change the infectivity of the virus. Oh, sorry, I was looking at the default plot. And here, let me put this to a more reasonable value so that spreads out a bit more. And once again, this is going to make that curve steeper as the virus has an easier time infecting cells. If we change the infected cell death rate and make it higher, we see a steeper decline in the virus, which once again makes sense because the presence of infected cells is what drives that amount. And similarly, changing the clearance rate is going to really impact that curve, since that value is going to impact. It's difficult here, right? Because we only see these arrows, but really this is a closed loop diagram. So they replicate the paper very well. And then they also talk about R naught here, and they frame it as a parameter. So basically they had the derivation here, and they cite the source that they're using for that. And what you can do is that you can change the value of this. And they have, I wish they had this for all the plots at points, but you can also change the number of days that it's simulated, so you can look at the resolution depending on how far you stretch things. Because you can see that if you have R naught set really low, you have to stretch out a little further to see the equilibrium behavior. And then this is a repeat, I'm pretty sure, of looking at, okay, yeah, this is looking at the model's fit to the data from up above. So now you can see that for the no model delay, based on the data sets, these are what we get for each patient's data. And they have this fun little animation where they show how they did that parameter estimation. So you can see it with the chi-square test going through. It takes a little bit, so we don't need to watch the full thing, but it's there if you want to look at it. Eden, do you want to take over for the next one? Okay, I'm going to mute myself.
Scroll. Yeah, okay. Just go down a little bit. Well, you can't see the equations and the figure. So in the second model, it's adapted from the first one to include a second class of infected cell and also interferons. So the intuition here is this is very similar, except that infected cells can either be spreading virons or not. So they can sort of be expressing the virus or they cannot be expressing the virus. Which is shown between the difference between I1 and I2. So I1 is not expressing, I2 is expressing. And what you see in terms of F, that's this concentration of interferons. The idea here is that some of the infected cells, the expressing cells can release these interferons, which reduce the transition from infected to infected and expressing, shown by the bar going to KL1, and they can also reduce the amount of viron put into the system, shown with the line going to PL2. The interferons also have their own decay rate, and the infected cells only die at stage 2. So you won't get infected cells at stage 1 dying, in the same way that the source cells never die in this model either. Otherwise, it's pretty similar. Connor, if you scroll down, you can see now we've made the system 5-dimensional instead of 3-dimensional. And they show their derivations of K and P here, which are constants from before, that they're sort of altering to account for that interferon amount, which is shown in the denominator of both. And this is also, again, from the same paper from before. And once again, if you go down a little bit, you can see one thing that I liked about this program is they offered really detailed instructions of what to try and what it meant, like what to intuit from it, which is good considering that like now we're looking at a system of 5 equations and a bunch of parameters, and it could be pretty overwhelming to sort of just dump that and not say anything about it. And in fact, you can even see at the bottom, it gives pairs of parameters to change together, which I think is really important. Now, unfortunately, oh, and this is again the table. Unfortunately, this one's the broken one. So it turns out that the last 3 here are completely mislabeled, which is, so you can see, like Connor, if you increase, you can see, actually, you can see it without increasing anything. You can see, for example, the last one, it says rate of IFN degradation is 0.7. The last one. Oh, yeah. And then if you go down to the output of the model, you get delay of IFN response is 0.7. So it's sort of, we had a harder time playing with this one, and actually it is funny how much not having that one-to-one match makes it hard to logic out what's going on. But the first 4 work. So if you're, say, to increase the infectivity of the virus, the second one.
Yeah, we got it. Sorry, lag.
Is it changing? Change something else. Maybe you need to load it again.
Yeah, I'm gonna reload it.
This does usually work.
Yeah.
I promise the whole second plot isn't completely broken.
Delay. Let's try this again. There we go.
That's better, right? So infectivity basically speeds up almost all of the curves because they're all contingent on virus production. And again, like Connor said, it would be nice if this one had days, so you could also zoom in on the resolution here. But unfortunately, this one's standardized to 8. But it is also nice. They do a good job of showing you your parameters at the bottom, even though they're mislabeled. So yeah, you can keep going. No, I'll keep talking.
Okay.
Can you scroll? I think they have the treatment.
I do want to make one more comment about this. Sorry, I guess I know that there's a concern about there being too much going on a plot, but something that I'm noticing is that these are not crazy high dimensional systems, and every plot so far has left at least one of the state variables out of the time series. And it's usually a pretty important one. Like it's one that would, like, if you're trying to understand how these peaks are ordered, not knowing the population of the infected but not expressing cells matters, especially because that transition is one of the things that interferon functions on. And they did the same thing for the other plot. And it's not really clear to me that there was a good reason for leaving those variables out specifically. So that's all I want to add in. I will now keep scrolling.
No, that's a good point. Thank you. Once again, you've got individual patient data, which is same principle as what Connor showed you last time, just updated for to show the interferon amount in relation to the virus. As this is what's measurable. And I think that might be part of why certain things were left out, is because the original data, maybe something like the number of infected but non-expressing cells wasn't like measured in the paper, but not that that's a good reason, but.
No, I know. The reason I'm going to push back on that is that, like, let's say, okay, that is the reason. That's fine. That's the reason to not have a data kind of like curve on that in your experimental plots. But if part of the idea of models is to also try to predict other things that we don't currently have measures for, like, there's no reason to not plot the time series for that variable in the system. Like, we do have that information. And it would be interesting to see what that variable is doing, especially if the rest of the trajectories match the data pretty well.
Yeah, I agree. I think I'm trying to predict why they did it that way, but I can agree. Once again, you've got a fun model fitting video. I really appreciated the combination of the data from the papers and the modeling in this case. I think the closing of that loop isn't something I get to see super often when looking at modeling literature myself. So
I don't think those are videos. I think it's actually doing the fit.
Oh.
That's another case where it probably would have been nice to see the code if we didn't know that that was something we could do.
So if you hit edit app, you should be able to see the code.
Okay, let's see. Yeah. All right, cool. Do you want me to keep no, wait, we're not gonna be able to load stuff. Let me go back to it as an app so I don't need to load the entire Jupyter notebook.
Yeah.
Okay.
I'll keep that a bit.
Too far. There we go. Okay. Okay, there we go.
Yeah. And so the next thing they did in this case is they looked at the delay model with an antiviral treatment, and they have the reference for where they're deriving that idea from, but they really don't say a lot about it. It's kind of completely black box. There's no equations or anything here. It's just sort of showing. I'm pretty sure what you're looking at here is the viral load, which is sort of that original patient data, and then what happens if they take an antiviral. And I think we can intuit that it makes there's less virus in the system, but it's hard to say at what point in that network is the pressure being put on by the antiviral, which would have been nice. So I'm not exact. This was sort of proof of concept-y, I guess. I think it could have been fleshed out more. It is pretty cool, and yeah, you can keep going. I can do this too.
Oh, it's okay.
So in this case, this is the third model that they provide. And so this reverts back to that first. There's no delay in this one. So there's no interferons. There's only one type of infected cell. The addition here is that you have another cell type E, which are these effector cells, which are supposed to model going in and having cells that kill the infected cells before they're able to release virus into the system, or while they're releasing virus into the system. So you can see it's the same diagram as the first case, except you also have this effector cell, whose equation is governed by the amount of infected cells. It has its own death rate in interaction with itself, and it has a death rate in interaction with infected cells. So it is both. And once again, the original healthy cells have no death rate associated with them. And so now there's 4 equations in this case. It's the same 3 as the first one, and then you have this DEDT, which tells you about the effector cells. And once again, this comes from a second paper as opposed to the original one that most of the modeling has come from. So in this case, they don't have any patient data. This is more of a hypothesis of the mechanism behind what's going on in the first couple models, if we explicitly define this other cell type. And once again, you get a very similar sort of graph where here we can play. And once again, the variables are a little mixed up here, it looks like, but I think the number assignments are actually right, which they weren't in the previous one. So the output doesn't read according to the sliders, but the sliders still work. Do you want to say something?
No, I'm just admiring that they managed to get things mixed up on the output and the input side twice, because I mean, it's also like, I think this one seems like it's possible. It's more wrong in terms of the naming of like what they had printed here. Whereas the other one, the reason we figured out something was off is because the outputs were looking irregular for what we were varying. But at the same time, as an application, my instinct is not to trust anything here until I look at the code and make sure that these are actually connecting the way the equation suggests they do, because the output looks different. Yeah, that's all.
Right. So in this case, I mean, Connor already did it, but when you pull the first slider, you can sort of see the effectiveness of these effector cells on the infected cells. And you can see basically the viral load is the peak of the viral load is significantly delayed. And if you increase the length of the simulation, you can sort of see the delay a little better, right? Yeah, so that's a good example of that. They have a calculation of R0 too that they output there. And then similar to some of the other models that we've seen so far, we've got a nice reference list without much discussion of anything else. And so some of the things that I actually really appreciated sort of the direction of the application. It did a really good job providing useful exercises to sort of at least get your grip on what the models were doing before you sort of played with them freely. I will say that Connor and I are lucky to have a bit of math background because none of the math is motivated at all. It's more sort of punted to the paper that's deriving it and sort of and letting the user go there to sort out why. Which there's some trust involved in that. So some pros and cons. Obviously, a couple things are broken, which is unfortunate because I think actually like there's the code's not buggy, it's just labeling issues. So which is enough of an increase on mental load to make it impossible for me to parse some of what's going on. But yeah, Connor, other thoughts?
No, that basically covers it. I do want to say that as a whole, I think the organization of this as an application was actually really wonderful. I kind of liked being able to jump from model to model and see how different factors were included. And I think that for the parts where they did go into the effort to include writing and discussion, those parts were very thorough. The few points where they just kind of like left you with a sentence of like, here's a graph, you have slider bars, have fun, was like an exception compared to what otherwise was a pretty good job. I think that this is the type of thing that, like, if I had to point someone to like what a good nanoHUB applications looked like if that code gets fixed, I would be willing to point people to this app. It's just that some kinks need to be worked out first.
Okay, other comments or questions? Man, I think
Hayden had a question in
Go
the
ahead.
chat.
Sure, go ahead.
I just put the title in there.
Okay, great. Thanks. Yeah, I just wanted to take a look.
I mean, I think so far one of the things that's been interesting is every one of these has had some strengths and some weaknesses. And I think one of the other apps we saw so far also had some data fitting, but this is the first. This one probably does more fitting in the app than the others that we've looked at, which is interesting. There is a theme which I think I'll see if I could punt to Giuliano, which is the laying out multiple plots at the same line in the same row requires a little bit of matplotlib fussiness, and so we should probably have some boilerplate code that lets people do that and distribute that to everybody. And also I should probably include a few slides in a lecture about it, just so that if people want to do layouts where you have 3 plots next to each other instead of on top of each other, you see how to do that. It was clearly enough of a pain that most people didn't do it. That to me already says that it's something that should be included in the course material. Yeah, there you are, Joel.
Yeah, there's a little. Yeah, I put there and didn't explain anything, right? So my bad. Yeah, the first number there is the x axis. It's the y-axis of the figure, and the second one is the x axis. So if you say 1, 2, it means you have one by the side of the other one. Right, and you establish the figure size. I forgot what DPI stands for.
Spatial resolution probably.
Yeah, it is.
Yeah, anyway, and then you say which one. In this case, you say this is the first plot and this will be the second plot. That's how you target them. But you do the same thing. Instead of you saying plot PLT dot and you do the whole thing, you do the axis instead.
Well, maybe you should work, if you're willing, you could work up a few exercises on that, maybe even a couple of slides that you could show to everybody.
Sure.
Because it's not trivial to figure it out. I remember going to the matplotlib documentation, trying to template off of that, and finding that it took quite a bit of effort because they didn't have the example. The examples they gave were not close enough to the ones I wanted to do, that it was easy to figure out how to change the examples in the manual to give you what you want.
Yeah, if anyone else has experience with matplotlib and want to jump in and help me, I am accepting too.
I mean, it's not dramatically complicated, but it is easy to get it wrong. And debugging it is not trivial, at least in my experience. Other comments? I think again, everybody's. So far, nobody came up with one of these examples where they said this was worthless, which is good. I tried to pick ones that I thought would be of interest to people, and I realized that there was a lot of infectious disease modeling. On the other hand, we had 2 years where the course was built around infectious disease modeling. So that's I know that wasn't people's interest this year, but I thought some of the lessons from it could still be interesting. So let's see, Gabriel, did you want to present something?
Hi, yeah, mine was actually Zach's presentation. I was just chasing my dog during it.
Okay, so you were together, all right.
Yeah, sorry if I didn't make that more clear. I thought I had mentioned it, but yeah.
No, I knew Connor wasn't going to be there, but I didn't catch everybody's.
So
yeah, no, it was Gabe's dog.
Yeah, the three of us, Eden, Gabe, and I lived together, and literally at like 3:50, we realized the dog was gone. So we were all sprinting throughout the neighborhood for an hour, which is why the three of us came in late. Apologies.
No, it's okay. I hope you were able to rescue the dog.
We found him.
Yeah, yeah. It was thanks to some person found him and they put an ad out. What was it, Eden? On like Facebook or something? And
yeah, next door. I don't know.
Next door,
yeah.
There's also Facebook has a page like Bloomington News that will do that. We lost a chicken and posted there. Somebody found a chicken, but it wasn't our chicken. We never did get our chicken back.
That's unfortunate.
Somebody stole our chicken. I can't imagine why anyone would steal a chicken. They did.
Must have been out of eggs.
Or it could have been a fox.
No, because somebody opened the cage, took out a chicken, and then closed the cage again.
Well, there is there's a whole thing where if you park rangers will tell you that if you ask them why bear-proof trash cans don't seem to work, they'll say it's because there's a considerable overlap between the smartest bears and the dumbest humans. So it's really hard to find something kind of in between there. So maybe you just encountered a very, very smart animal, but either way, that's unfortunate. I'm sorry.
Well, it's a while ago, but I'm glad you were able to recover the dog because that's very stressful. Well, that's great. I hope this was helpful. I'm not sure how to use the rest. We have 40 minutes left. I'm not sure what the best way to use the rest of our time is. I mean, do people want to go through their outlines for their projects, or would people prefer to save that till next week and have a little bit more class lecture type class for the rest of the class?
Can I ask a question?
We've done a lot of talking already, so I don't mean to put you on the spot either way. But if you're ready to go for your presentations, I don't want to prevent you from doing that either. Go ahead, Joe.
Yeah, I just was thinking about what is the difference between, because you mentioned you were gonna talk about Michaelis again and Hill function. And since I was working on the Komarova-Fang models, I was wondering what is the difference of just doing an inhibition of 1 minus the number of cells and using Michaelis in that case?
Oh,
or in similar cases?
Well, that's an important one. And for what you're doing, you may need to use 1 minus.
Okay.
So the question was, if you're doing inhibition, and it's probably easier to do it if I had a slide up on it. You can have in inhibition, you expect the rate of something to decrease as the amount of your control substance goes up. And you could imagine that the simplest thing would be a linear decrease in the rate as a function of the amount. And then you have to ask the question, when you get to some threshold, the rate will go to 0 if it's a linear decrease. And the question is, does the rate go negative as you keep increasing the parameter or not? And for population control models, very often they work best if the rate does go negative. Now, a rate of chemical reaction, what would it mean if the rate of chemical reaction went negative? It would mean that you had the back reaction happening more faster than the forward reaction. In the case of something like the rate of replication of a cell, it doesn't make sense for the rate of replication to be negative. On the other hand, if it's the total rate of growth or shrinkage of the population, that's the birth rate minus the death rate. And the total rate of growth of the population could go negative. So you have to ask really at a fundamental level what the regulation means. Is that rate law defining something that has to be positive, like the rate of proliferation of cells? Or is it actually representing a difference between two things, like the difference between births and deaths, the net rate of growth of the cells, which could go negative? And in those very simple regulatory models, like the ones you showed in your plan, really you have both. If there's no explicit death rate included for the cells, then you have to include that in those rates. Otherwise, you'd have this came up in one of the examples where somebody left out the death rate of a population, and that does fundamentally change the dynamics. And so, you may find that if the values aren't allowed to go negative when you overshoot, that you can't get a stable equilibrium.
Yeah, I think I thought about that because of homework 5, we play around a little bit with that. And then I was like, well, it's the same effect, but I don't know, what does one, is doing that is too different from the other. But thank you.
I wanted to come back, just remind you of a few of the things last time. We talked about loading and running SPML models from the biomodel database, how to find them, a little bit about how that database worked, and a little bit about how to convert them into antimony and run them. And I hope that was useful for people. And then we defined the Michaelis-Menten approximation to enzymatic reactions, introduced a key concept, which was the Michaelis K, which is the concentration of the substrate at which the reaction has half its maximal rate. And that half maximal concentration is one that comes up also for Hill equations. And so it's pretty important. A concept of a maximum velocity for the reaction, and then the ability to write the Michaelis-Menten approximation either in dimensional form, S over Km plus S, or we could write exactly the same thing where we divide by K over K, and then you express S as a fraction of Km. So then the concentration relative to the half maximal concentration. And this came up a little bit in one of the questions that was discussed about the project, which was whether it's more useful to plot variables by their absolute values or rescaled so that you have them all at the same range. And this is something that we have to think about case by case. I don't think there's a universal answer to that. This does give us a sense of what it means to say that a concentration is large, small, or intermediate. In this case, if a concentration is much less than the Michaelis value, the concentration is small. it's much bigger than the Michaelis value, it's large. If it's roughly equal to it, it's intermediate. And so we now know that if we want to explore small, medium, and large ranges, what the value of the variable has to be. And we showed that the Michaelis approximation works pretty well in a lot of cases, but it doesn't give us that short-term initial situation when the enzyme and the substrate are forming its initial complex. It doesn't work, of course, if you have long chains of reactions or cooperativity. And it's limited in the sense that you pretty much need the amount of substrate to be much larger than the amount of enzyme for the thing to work. And then we did the same derivation a second time, but instead of the situation where the enzyme bound to one molecule of substrate, we imagined the enzyme bound to multiple molecules of substrate simultaneously. Or you could imagine that being a stack of reactions where the enzyme binds one substrate and then a second one, and then a third one, and then a fourth one. In either case, you wind up with a Hill constant that's structurally identical to the Michaelis constant. It's the half maximal activity concentration. But here there's this issue that the concentration is raised to the nth power. And so you have to decide whether you're going to use the nth root of that Hill constant or not. The one that usually is easier to use is the one that has units of concentration, because you could then scale the concentration the way we did before. Total amount of enzyme hasn't changed, the Vmax hasn't changed. And so we can then write the Hill rate law again in scaled form, where we divide by K, or in the unscaled form, where instead of K, we have a Kn to the n in the denominator. And sometimes it's easier to abstract this as a Hill function, where you say there's f of n is x to the n over 0.5 to the n plus x to the n. That's effectively equivalent to the scaled form of the Hill rate law. But here we'd be scaling by 2k instead of by k. And again, one of the values of having that k is it gives us a sense of what it means to say the concentration of S is small, medium, or large. We see our stack of Hill functions as we change the exponent. Exponent of 1 gives us Michaelis, and that's fundamentally different from everything else. The value is always a half at k. It always saturates, goes to a constant value for very large values of S. But in one case, the slope is maximal at 0. In the other case, the slope is maximal at k. And the steepness of the transition increases as the value of n goes up. And so this allows us to do progressively more and more step-like transitions, which may represent different biological situations. In both the Michaelis case and the Hill case, if we take the numerator and replace the concentration of our chemical species by our k, we get a decreasing function. It's the counterpart of the increasing function. And so we automatically, when we write these functions, have an increasing Michaelis function, decreasing Michaelis function, increasing Hill function, decreasing Hill function. This kind of method is very useful when we want to model signaling in regulatory networks. We're typically going to model the effects of activators or inhibitors as Hill functions which modify the velocity of the reaction. In reality, these interactions could be quite complex, but we're typically going to simplify things and embody our ignorance in the form of a Hill function. If we come back to the Hill function for a second, we'll notice that the Hill function has 3 parameters. it has Vmax, it has K, and it has N. And we can usually rescale the time scale if we wanted to get rid of the Vmax, or we could imagine that K is rescaling all of our variables, in which case the K would disappear. And so really there are two fundamentally different parameters in a Hill function. Three, depending on the relative scales of the system. And so it's nice because even though there's a lot of possible complexity here, we could reduce the number of parameters we have to use to describe that complexity to 2 or 3. So I showed these slides very early on in the class, but they probably weren't very meaningful when I showed them the first time. And I want to come back to these now. And so what we're going to do is we're going to start out with a diagram that looks like the one on the left here. A activates S2. And we want to know what we do with that when we see it in a reaction diagram, in a network diagram. And if we have no information at all about the system other than that there is activation, we have to make some hypotheses about it. And there is a typical standard set of hypotheses that we're going to make. So we're going to assume that A activates S2, and we're going to interpret this in the following way. In particular, this doesn't tell us what is being turned into S2. It says that S2 is appearing out of nothing. So the only thing we can do as a chemical reaction is to say nothing goes to S2. The rate of that reaction depends on A. And so when we see A activates S2, we're going to interpret this as nothing goes to S2 at a rate that has some Vmax, a maximum rate of production of S2, times A to the N over K to the N plus A to the N. So it has a Hill form. Now, what the values of K, Vmax, and N are not described. Vmax will determine the time constant of this reaction. If Vmax is very large, then things happen quickly. If Vmax is small, then they happen slower. Notice it does not change the amount, the rate. The Vmax doesn't affect the dependence on the concentration of A. The only thing that does that is K. And so we have separate control over the level at which we go from not producing S2 to producing S2. If A is less than K, we produce very little S2. If A is bigger than K, we produce a lot of S2. The rate of production is given by Vmax. If we saw the diagram S1 goes to S2, and that is regulated by A, promoted by A, then we will assume to begin with that we have either mass action, in which case with no regulation, the rate of reaction would just be Vmax times S1, or perhaps a Michaelis saturation of the reaction, Vmax S1 over Km plus S1, in either case multiplied by the regulator, A to the N over K to the N plus A to the N. And so this is actually pretty powerful, and it works surprisingly well. You can start out with a picture like this with no information about rates or functional forms. You can impose this set of assumptions, and I won't promise that it works all the time, but pretty often it gives you something that's biologically not bad, especially if you get the time constants about right. You can ask what values of K should you use. If you really don't know anything about concentrations, the one thing that you can say is that biologically, if A is going to regulate this reaction, then you expect that the value of A in the experiment will sometimes be less than K and sometimes be bigger than K. Because if the level of A were always less than K, the reaction would always be turned off. If the level of A were always bigger than K, the reaction would always be turned on. The regulation wouldn't do anything. And so it's not unreasonable to assume, for example, if you don't know anything about A and K, that K is a half and A goes between 0 and 1. So you're going to by essentially rescale A by K to begin with. The advantage also of keeping all of your concentrations between 0 and 1, or 0 and 2, or 0 and 10, is that just the way in digital electronics, in old TTL circuits, 0 was 0 volts, 1 was 5 volts. And that meant that you could combine the output of one circuit into the input of another without having to do a lot of resistors and adjustment of values. So if you rescale your parameter, your variables always by K, and you say we know that in reality some Ks are 10 micromolar, some Ks are 4 picomolars, and so on. But we know that the concentrations are going to be a few times K, K divided by 3 or 4. We're going to rescale all of our concentrations by their relative Ks. And then we'll say we're going to just set K equal to half for everything. Then we basically created a situation where the output of one of our variables can be used as the input for something else because it's all scaled to the same range. We may still have to do a little bit of hand rescaling in certain situations. But in general, if we force everything to be scaled with respect to its K value, it allows us to build circuits that work without having to worry about impedances between them. So for inhibitors, it's going to be the same thing. We see R inhibits S2. S2 has to come from somewhere, so nothing goes to S2. We write the inhibition as the inhibitory Michaelis function V max K to the N over K to the N plus R to the N. Again, very symmetrical. We've just changed one symbol, which is in the numerator of our equation. If S1 is going to S2 and it's inhibited, again, we'll write either Michaelis, Menten, or mass action for the reaction by itself, and then multiply the rate by the inhibitor. For both activation and inhibition, we might want to assume that with our inhibitor, even if there is a lot of inhibitor, the reaction rate doesn't go to 0, in which case we'd have to add a number to our Michaelis, to our Hill equation. So it didn't go to 0 when R got very large. And similarly for activator, we might want to assume that there's some what's called basal rate, so that we would add a number again to the functional form, so that you didn't turn off when you had no activator. I've simplified things a little bit here by doing that. That would correspond in electronics to a leakage current. If you're doing TTL logic, you don't want a leakage current. If you're doing analog circuits, then that's a useful thing. If we have a gene regulatory network, and here I have written those out explicitly, we'll see again, P1 promotes P2. In this case, the math is the same, but our interpretation of the biology is a little bit different. In this case, P1 is binding to a promoter region for the gene. The gene is being transcribed. That transcript is then being translated. So there are multiple stages of biology going on here through the fundamental dogma of biology. But again, our bottom line is that we interpret P1 activates P2 as nothing goes to P2. And here I've included explicitly a basal rate of transcription and translation. Even if there's no P2, excuse me, no P1, we still expect to get a little bit of the gene produced. Similarly, for inhibition, even if there is a lot of the inhibitor, you expect some of that gene to be produced. And those basal rates can be pretty important. They tend to make things fuzzier. They tend to make things a little bit harder to interpret, but biologically, those can be fairly important. But again, the value of K is critical. It's the concentration of the promoter or the inhibitor at which the transcription rate is half of its maximal rate. In the case of gene regulatory networks, genes typically turn on or off pretty sharply as a function of the concentration of the promoter or the inhibitor. And so the exponent that we use tends to be pretty big. The sort of default guess is 4 or 8. If you look in Herbert's textbook, where he has a sort of a library of possible functions, you'll see typically 4 and 8s in the exponents. Those don't really represent actual biological cooperativity. It's not that you have 4 copies of the promoter binding or the transcription factor binding. It's just reflecting the functional relations that are observed experimentally in terms of those translation rates. Occasionally, you'll see higher numbers. You'll see 12, 16, 20. It's not always clear where those come from biologically. Okay. Inhibitors behave the same way. And so we can do exactly the same thing. So I wanted to talk the last few minutes, maybe we're going to run out of time, a little bit about some of the questions that came up in class in talking about the exercises. And this is a hard one. In the homeworks, I'll often ask you, what do you see? And that's a sort of an open-ended question, and it can be a little bit intimidating, I think, sometimes. But there were a couple of people who pointed out that in some of the examples that were given, the user was asked to change a parameter value, see what happened, but wasn't given guidance about what to be looking for. As an example, there was the question, well, what rate of vaccination or what vaccine efficacy would be needed to eliminate the virus? Would be a kind of question that one might want to ask. And so I think exploring those exercises, those apps, can give you a sense of what are good questions. Now, very often until you've worked with the model and explored it, it's hard to come up with good questions. In other words, it's hard until you see how the time series change or how the dependence on a parameter changes. It's hard to come up with good meaningful questions. What did you learn from this exercise? But ultimately, if you're going to be doing mathematical biology, or frankly, any kind of biology, experimental as well, the key thing is that you're going to be trying to test hypotheses or come up with ways of learning about the fundamental behavior of a system. And so trying to think about what kinds of questions that you can ask and solve with models, I think is hard, but necessary. And I don't really have a simple answer because it is problem dependent. But I did want to walk you through some examples of possible kinds of questions you can begin with. These are not necessarily the kinds of questions that will extract meaning. Meaning is another level of complexity. In other words, and that's why I brought up some of the issues in the case of, say, tuberculosis. Wanting to know what happens when you have vaccines of different efficacy isn't so interesting a question when you have a vaccine with 98% efficacy. But when you have a vaccine with 20% efficacy, you could ask the question, is it even worth using it? Similarly, if you have a drug, you take the drug once and it cures you, then asking the question, what happens if people don't take the drug the way they're supposed to, is pointless. But in the case of tuberculosis, where you have an antibiotic that has to be taken for a year or more, and then it'll make you sick, and where you feel better after 2 weeks, convincing people to take that drug for a year is pretty hard. This can happen with medications for chronic infections like HIV as well. The antiretrovirals are pretty harsh. They make people feel pretty sick. They can damage your liver. And so there may be strong motivation or reasons to not want to take the drug. Sometimes actually with HIV, there's some evidence that actually going on cyclical treatment can be a good thing rather than a bad thing. But then you have a whole series of questions that come about how people interact with the situation, about the social consequences. And you also can have interesting questions where the results for the individual and the results for the community may not be the same. The interests of the community and the individual may not be aligned. Clearly, the math by itself doesn't tell you what those questions are, but exploring the math can help you answer those questions as you begin to frame them. And so if we think about a very simple network, we might want to design a network just to do some very simple things. And we're going to come back to this in the context of this simple feed-forward network that we're going to be looking at now for a week or so in the homework as well. Suppose we want to build a little biological network, like a gene regulatory network, that we want to have turn on when a signal is between two levels, but it turns off when this level is very low or very high. We clearly can't do that with a single regulation. An activator can't do that by itself, and an inhibitor can't do that by itself. What if we wanted the network to, as we turn it on, it makes a pulse of the chemical and then stops? That clearly also can't be done by a single activator or a single inhibitor. What if we want our network to switch from being off to being on as a result of our input, but then not care about the input anymore, to be a latch? That also can't be done by a single activator or a single inhibitor. And so all three of those things need you to be able to design circuits. And the homework that was originally due last week and was due today explores some of the circuits to do those things. One of the ways to begin to think about how to design circuits is to separate your thinking into the transformation of inputs to outputs generically, and then also to split your thinking into two parts, which came up also in the discussions today. The first one is steady-state behaviors, or long-term behaviors, because you may have an oscillatory behavior, chaotic behavior instead of steady-state. But fundamentally, what happens at long times? And that's what we did when we looked at Michaelis-Menten and Hill. We assumed that we were in a regime where time wasn't important anymore, even though we knew at short times it was, and potentially at very long times it was too. So look at, as a general rule, it's a good idea to look at steady-state behaviors or long-time behaviors first, and then to come back and ask, what are the short-time behaviors? What are the temporal behaviors? So those are some of the things that we think about. The second thing that we might want to think about, and it came up in the example where there was a comparison of death rate with and without treatment, is that biology thrives on differential measurements. To some extent, this is due to the fact that it was hard to make quantitative measurements biologically. To some extent, it was due to difficulty in repeating experiments. It's very hard to quantify the exact amount of a protein in a cell. It's hard to measure it in time. It's hard to get two cells to do the same thing twice. It's very hard to get two animals to do the same thing, if you do the same thing to them. And so one of the key concepts in biological measurement is that you'd make differential measurements. Instead of saying the amount of chemical X is such and such under this condition, the amount of chemical X is such and such under that condition, you do a differential measurement where you make the change and you look for the fold change. When I applied promoter X, the amount of chemical Y went up by a factor of 10. That has the advantage that I don't actually have to know exactly how much of the chemical I have. And so these kinds of differential measurements are classic in biology. They often are problematic because the same perturbation that is changing the thing that you want to change may change a lot of things you don't want to change. In physics, you try to do things that are orthogonal. You change the temperature, but you don't change the pressure. You change the magnetic field, but you don't change the electric field. In biology, it's very hard to change one thing at a time. But at least as a principle, this is something we do all the time. We turn gene X off, we turn gene X on, we add chemical Y, and we compare the output with and without that perturbation. And so the little feed-forward network that we're going to work on is very simple. It's in a sense the simplest kind of thing that we could do with this. We'll imagine that we have three elements, P1, P2, and P3. P1 does something to P2. It could be an activator of P2, it could be an inhibitor of P2. P2 does something to P3. It activates or inhibits P3. But the key thing that's a little bit different from what we've done before is that P1 also will inhibit or activate P3. And so now P1 has an effect on P3 directly, and it also has an effect indirectly through P2. And this is going to be able to produce not an infinite range of behaviors, but a much richer range of behaviors than just having P1 affect P3 directly. In particular, the time scale over which P1 affects P3 indirectly could be slower or faster than the rate at which it affects it directly. That means that we can have interesting temporal behavior as well as long-term behavior. And so this is a framework that we can use to begin to think about these problems. This is called the feed-forward network because P3 doesn't come back and affect P1 or P2. In all of the cases of interest that we've seen today, well, not all of them, but most of the cases of interest that we've seen today, there's some feedback. P3 produces virus and the virus infects P1. There is a regulation of the differentiation or rate of division of P1 by P3 or P2. And so that feedback gives us much richer and more complex set of possible behaviors than we have just with feed-forward. But even a feed-forward network by itself is able to do a reasonably large number of things. And chapter 14 in Herbert's textbook analyzes this feed-forward network in some detail. I have a lot of lecture material on it. I'm probably going to cut back on that, but we'll do a little bit of that. And so, boy, I'm out of time. I was hoping to talk today a little bit about the kinds of questions we can ask about network models. You've seen this picture a lot, and I'll just discuss that one more time because I think on the one hand, it's a very simple picture. On the other hand, there's a lot of meaning in it. And I hope that each time we see it, as we work through the class, the degree to which it's meaningful will increase. So a model is something that has a model structure. And that model structure in this case would consist of the network connections and the rate laws that we have. Are they Hill functions? Are they something else? Are they activators? Are they inhibitors? Connection. And then a set of parameters. And those parameters could be the initial values of our concentrations. They could be the rate constants. They could be those Michaelis or Hill constants and so on, the Vmaxes. Together, those model structures and input parameters will give us some time series, which is how the system evolves. And that time series may have some long-term behaviors, steady states or otherwise, that we're interested in. And so what we're going to do next time is come back and just look at some very simple kinds of phenomenological questions we can ask about those behaviors. And let me just give you a taste of that here. The model structure and the interaction diagram and its parameters will give us a series of time series of concentrations of species, or some derived quantity, the sum of them, the square root of them, something else. There are a limited number of questions, types of question we can ask directly. This is now not talking about meaning, like what level of vaccination do I have to give to get rid of my virus in my population? Although that could be a question. But there are a limited number of things we can ask about time series of scalar values and how they depend on input parameters and model structure. Which questions we ask are going to be significant in different cases. And let me just do the first one, which is we can ask very qualitative questions. And these are questions that don't refer to specific numerical values, but to categories of behavior. And we've seen this already today. Qualitative questions can be useful because we may not be able to measure values quantitatively. We may not know how many cells there are. We may not know the concentration versus time. They're also more robust to model approximations. The exact value may depend very sensitively on what we picked, whether the thing goes up, down, or whether the thing oscillates or not, may not. And so if we have a time series, there are not that many qualitative outcomes we can have. We can reach a steady state, which is what we see most of the time. We can have periodic oscillations, which we've seen, some of the examples we have. We can have chaotic time series, or we can have divergence. The concentration or value could blow up. That's really all that can happen to a time series. If it's bounded, it either reaches a steady value, it oscillates, or it oscillates aperiodically. Can't do anything else. If it diverges, and we can't say much more about it than that. And divergence biologically is usually death. And so we can ask questions like, for a given model with a given set of parameters and boundary conditions, initial conditions, which of these four things happens? And that gives us a series of interesting questions because we might want to know how many states of each type are there? Are there multiple steady states? Are there multiple oscillatory states? Are there multiple chaotic or divergent states? And we'd like to know about the boundaries between them. As we change a parameter, do we go from a steady state to an oscillation, from an oscillation to chaotic behavior, from a chaotic behavior to a divergent behavior, or something else? And these are typically very important because they're going to describe things like differentiation, movement, cell division, death, birth. And so if we're able to describe the qualitative behaviors of a system as a function of the parameters in our model, we have understood a lot about how that model works. And so the fundamental thing that we talk about there is what's called the phase diagram. The pattern of behaviors as a function of functions, as a function of the parameter. So for example, here, imagine I've got a neuron, and I have the potassium concentration on the x-axis and the sodium concentration on the y-axis. And I say that if the potassium and sodium are both low, or potassium is low by itself, or sodium is low by itself, we get a steady state, or system equilibrium. For some intermediate amount of sodium and potassium, we get periodic oscillation. If we keep the potassium concentration intermediate, but increase the sodium concentration too much, we get chaotic oscillation. And if we make our potassium level too high, the system diverges and our cell dies. And so this, as a fantasy diagram, here it's about cardiomyocyte, sorry, not about a neuron, about how that behavior of that muscle cell might depend on ionic concentration. We did a little bit of that game when we said, what if we change the glucose level of outside of an E. coli? How fast does it grow? That would be a one-dimensional version of this. This kind of diagram, phase diagram, is a very, very powerful way of encapsulating information. And we might expect that if our model is right, the identity of these domains and their relative positions matter. We might expect that if our model, things are squeezed vertically, stretched, twisted a little bit, but if we have steady state oscillating chaotic diverging in a particular sequence, if our model gets that sequence right, we don't care if the exact positions of the boundaries are right. And so this creation of a diagram of this kind gives us a very, very powerful handle on how our systems behave. It tells us what's possible in our system. And I think that I want to stop with that. I've run over time a little bit, but I want to stop with that, and I want to leave you to think about that in the context of the questions you're asking, both in the homework problems and for the projects you're working on, and as a way of organizing your thinking about all of the projects you work on. Now, if you're working with Randy Beer, you've probably heard this kind of thing put forward a lot. Depending on your background and experience, this may be a very familiar set of concepts, but it's something that, again, I think tends not to be taught as explicitly in many places as it could be. And I hope, again, I think if you bring this up too early, it seems too abstract. But now that we've seen it in action in some of these examples and homework problems, I hope it's more meaningful. Are there any questions or comments about this? Because I think this is where I wanted to stop for today in terms of the lecture. But I want to bring it up and let people talk about it, because there's a lot to think about when you address these kinds of problems.